Customers are very happy about it. We have repeat customers coming back for us, asking for usage of this system for other molecules which they have in their pipeline as well. So as a CRO, I would believe that this system is a very good system to have. Although we have used multiple other systems in our previous experiences, but this system gives us a lot of robustness and versatility, which we have not seen in the industry in the last few years. So my name is Dr. Sanjib Banerjee. I take care of the biopharma business for Veeda Clinical Research. To give you a bit of idea about what Veeda Clinical Research does, it's a clinical CRO which had been working almost like 20 years on different clinical studies, including healthy volunteer subject studies and clinical trials for different modalities. Recently, it has acquired one of the largest clinical research organizations in Europe, which is called HEADS. Apart from the clinical research activities which Veeda does, Veeda is also engaged into preclinical services through a group company called Bioneeds. I represent a group over here which takes care of the biopharma business, basically working on the molecules, not on the clinical aspects of the molecule, but the molecule themselves. And my role over here is to take care of the entire business of 4 different verticals which we have, where analytical characterization is one of the most important verticals we take care of for looking into the structural and the functional integrity and comparability between biosimilar molecules. So that's something which we do in Veeda Biopharma in Bangalore. As a part of our activity which is related to the biopharma business, that is, we work a lot on molecules which are like biosimilars, vaccines, and novel biologics. What we do is we try and understand a lot of these functional and structural aspects of these molecules when it comes to the efficacy of the molecule, as well as which is related to the clinical aspects of this molecule. So nowadays, with the biorationality law and the Biosecure Act putting in place, it becomes an essential element of a biosimilarity assessment, where people compare in between 2 molecules, one innovator and a biosimilar, and try to see the kind of similarity which exists as a part of their biocomparability package. Now, what we do in Veeda Biopharma with the Chirascan is that we use it extensively for understanding the second and the tertiary, or the secondary and the tertiary structures, more in between these 2 type of molecules, which is one of the innovator molecule, the other is the biosimilar molecule. We have put a lot of effort in getting this Chirascan over here, because we had scouted multiple different instruments for this process, and what we realized is this Chirascan which we have provides the largest and the best way of showing how the molecules are when it comes to the production, the screening, the degradation studies, the quality controls, and the biosimilar development aspects when it comes to the formulation development, or even the API molecule characterization itself. So that's what we do. Yes, definitely. I mean, with Chirascan, it has been a very good experience with us. I mean, we had been using this instrument quite often for different customer services, where the customer comes to us for a system which is highly sophisticated, and this system brings into the structure a lot of the sophistication which is required for doing this kind of biosimilarity assessment. We do have other instruments with us for secondary structures, and Chirascan fits very well with those structures when it comes to the FTIRs, or the other different way of looking into the secondary structure of a molecule. So Chirascan has been a success story for us. We had seen a lot of these molecules work out very well with the Chirascan system because of the different integral parts associated with the Chirascan. I mean, we really use the different temperature meltings to understand the fingerprint similarity in between molecules, because the other systems which we evaluated at point of time when we were looking into the Chirascan system did not have these kind of a single cell Peltier based system, where we can use it for the scanning across different temperatures for the secondary structure of a molecule. looking into Chirascan and applied photophysics was something where we started very early. We thought that, when we were setting up this lab, we were thinking about getting the state-of-art instruments with us. Now, what we realized that, there are different players in the market, but there are few of them which are totally focused on a certain kind of activity, and applied photophysics was one we thought that we would have, because, a lot of other instruments use the platforms which are developed by applied photophysics for their own systems. But looking into applied photophysics was one of the most important thing for us. This is not the only system which we got from something which is different from what exists in the industry. There are other systems also which we have got after doing multiple scouting and figuring out which is the right system for us to have a robust structure for doing this kind of activities which other CROs in India do not have. The important thing over here which we have to think about is that, there are multiple CROs with similar systems along with them, which do not always come up with the expected results which are in the market. So there have been multiple times where people have come to us, even though they have a CD system with them, they have come to us for using our Chirascan system for getting something which has not been sorted out by their own CD equipments which they have inside their labs. They have the best system as well as they know how to do it. So these are the two important things, because sometimes you really have the system with you, but you do not know how to maximally use this, or may harvest the system most possibly. Over here, what we have, we have the expertise, we have a very good support from the applied photophysics group, which also enhances our skill set by solving certain issues or problems which we face also while using the system. But then these are sophisticated system, and then if you can use it to the maximum, you'll always come up with better advantages than any other system which are there in the market at this moment. Yeah, I mean, the Chirascan system has been one of the champions in the lab. I mean, it has got us a lot of these projects because of the versatility of this instrument which is there. The best thing is the data which is generated over here is, say, suppose I'll give you an example over here, which is like the biosimilarity software which we have, right? A lot of these other systems do not have that kind of a software. So if you're talking about, different temperature melting points, overlaying them, showing the fingerprint similarity becomes much more easier over here, which you have to do manually somewhere else, some in other systems. So I mean, if I go with the the kind of output which I have got from the system, it is extremely good, and our customers are very happy about it. We have repeat customers coming back for us, asking for usage of this system for other molecules which they have in their pipeline as well. So as a CRO, I would believe that this system is a very good system to have. Although we have used multiple other systems in our previous experiences, but this system gives us a lot of robustness and versatility which we have not seen in the industry in the last few years. I mean, I think like in a CRO business where there is a lot of competition in between the different CROs which we have in India as well as outside India, it becomes a challenge to generate the kind of data which is acceptable to the regulators as well as comparable to the to my competitors outside India. As I see that this system is a quintessential system when it comes to the output it has, because it gives you that kind of a mobility where you can actually play around with a lot of different aspects of doing the circular dichroism, as well as figuring out how you can use this system for assessment of biosimilarity. It comes with a very good hardware, and then the software adds an advantage to it where a biosimilarity comparison can be done within a very short duration. That's something which every CRO should have as a part of their equipment list.